So you have this. So now, how many different ways in which this child can be generated? I mean, can have be. What are the different ways in which you could conceive this child using these 4 different genotypes of the parents? There are 4 different crosses you can do. They're all mutually exclusive. There's only one child. So you can have a big R father crossed with a big R mother. Or you can have a big R father crossed with a big R mother. Or you can have a big R father crossed to a big R mother. Or a big R father crossed to a big R mother. Right? So there are 4 different ways in which you can do this cross. Okay. So, grandparents' genotypes are known, parents' genotypes are partially known or unknown. You want to calculate the probability of having an affected child. So, father has a 50% chance of being big R, 50% chance big R, little r. Mother has a 1/3 chance of being big R, and a 2/3 chance of being big R, little r. So, there are 4 possible matings. In the first mating, there is a probability of this 1/2 crossed to the 1/3, right? But then what's the probability of an affected child in this cross? 0, right? Because it's a recessive trait. The next cross is basically a big R father crossed to a big R mother. So 1/2 crossed with 2/3. And what's the probability of an affected child again? 0. So once again, that's a 0. The third probability is this father, 1/2 big R crossed to big R mother, right? So that's 1/2 crossed with 1/3, but the probability of having an affected child again is 0. And then the last cross is a big R father crossed to a big R mother, so that's 1/2 crossed with 2/3, 1/2 times 2/3. And then the probability of an offspring in this cross, heterozygous to heterozygous, probability of homozygous recessive is 1/4. Correct? And so it's 1/2 times 2/3 times 1/4. Now you combine all of these 4 probabilities together because they're mutually exclusive. It's either this or this or this or this. So you have to add all of those conditional probabilities together. So 1/6 times 0 plus 1/3 times 0 plus 1/6 times 0 plus 1/3 times 1/4, which equals 1/4. So the probability of this child being affected in this cross is 1/4. Yeah? I'm going to let you digest this a little bit. If you have questions, ask. Go ahead. That's a dominant autosomal trait, and this is a recessive autosomal trait. Yes? Can we move on? All right. So that's pedigrees. So we have finished now with chapter 4, I believe. Yes, we finished with chapter 4. Now we're going to go into chapter 3, only the first half of chapter 3, 3-1. Okay. 3-2 comes after the midterm. So you want to focus on 3-1 for the midterm. That's where we're going next. It's only 8:35. Do you guys want to take a break now or later? Later? Okay. Okay. So until this point, you know notice we were looking at how genotypes and how phenotypes are passed on from parent to progeny. And we looked at genes on the X chromosome, and when they're on the X chromosome, they don't precisely follow Mendel's rules. The phenotypes tend to kind of fluctuate a little bit, correct? That was a variation on Mendel's rules. We're now going to continue along that path. We're going to look at other phenotypes that don't precisely follow Mendel's rules. We're going to look at phenotypes that are regulated by a single gene. Okay? So we're going to focus on single gene traits. One gene affects one phenotype, but they don't precisely follow Mendel's rules in terms of the phenotype. Genotypes, there's no problem right now. It's just the phenotype, the appearance that tends to not follow Mendel's rule. All right, so what is the biochemical explanation of a phenotype? I've already told you what a gene is. It's a segment of DNA that codes for a protein, correct? So imagine a protein is an enzyme. It takes a substrate and it converts it into a product, right? And the enzyme, that's the protein that moves the substrate and converts it into a product, and there is a gene that makes the enzyme. So there's your relationship that you can imagine. Okay. Now when we talk about dominance and recessive, what we are saying is the protein made from the gene with big A has a phenotype that is masking the phenotype of the protein made by the gene little a. Okay. And big A, and big A, little a have the same phenotype. So this big A protein is masking the presence of the little a somehow. So, what we talk about when we talk about recessive and dominant is we're looking at the heterozygous condition, correct? Everything is in the heterozygous condition. That's how you determine whether something is dominant or recessive. If it's just big A, you can't tell if the big A gene is dominant or recessive over the little a gene, other than the fact that we use it as a convention. At the level of the protein, you have no idea. The only way you figure that out is by looking in the heterozygous condition and asking which trait is the one you observe. So let's imagine something like height of a plant. It has a dominant allele, big T. And so when you have a condition, big T, the plant is 6 feet tall. When you have a heterozygous, big T, little t, the plant is 6 feet tall. And when you have little t, the plant is 2 feet tall. So based on this plant, you're going to say that 6 feet is the dominant phenotype and 2 feet is the recessive phenotype. Correct? Because it's in the heterozygous that you determine whether something is dominant or recessive. Imagine a situation where it's the opposite. Here's a situation. What if instead of this, a big T was 6 feet, a big T was 2 feet, and little t was 2 feet? What would you say? You would say that 2 feet, the phenotype 2 feet, is dominant over the phenotype 6 feet. You would ignore the uppercase and the lowercase letters. You would just focus on the phenotypes, right? And it would be the phenotype in the heterozygous that would basically be the basis of your definition of what is dominant. Fair enough? Okay. So now you would say that short is dominant to tall, not tall is dominant to short. So dominance and recessive are operational definitions. They are subjective definitions. They will change depending on the condition you are analyzing.
Is this the same as you say when something's haploinsufficient or haploinsufficient? Or
No, haplosufficiency and haploinsufficiency is something quite different. Okay. So don't go there.
Question?
Yeah.
In a heterozygous individual, are both of the alleles producing different enzyme proteins?
They're producing different forms of the same enzyme protein.
So they're both being made in the cell, but only the dominant one is functioning?
Correct.
Okay.
But what we're going to talk about now is instances where that doesn't. Both of them are being made, and they will both affect phenotype. But that's what we're going to do. But yes, you're absolutely correct. Under classic Mendelian definition, both proteins are being made, but only one form is what you observe. The other form is being masked. Yes. What it's making, you can't see by your naked eye. Okay, so as I said, genes are responsible for the production of specific proteins. Enzymes are proteins. So in a biochemical reaction, there is a substrate, there is a product, and there is an enzyme. So imagine an enzyme takes a substrate that's smooth, converts it into a product that's wrinkled, right? If you may have the enzyme present, you will make wrinkled seeds. If you don't have the enzyme present, you would make smooth seeds. I'm just making this up. I'm just giving you an example, right? So the gene for this enzyme makes this enzyme. There is a gene on the chromosome that will make RNA. That RNA will get converted into the protein, and that's the enzyme. So when you look at the phenotype, you're basically looking at the function of this gene. Correct? Now, what's the definition of a wild-type phenotype? It's the phenotype that's observed most of the time in nature. Now, what is nature? Is it the entire world? Is it the classroom? Is it my lab? That's your decision. You decide what the population you're studying is. But in that population, what is the most prevalent phenotype is the one you would call wild. Okay. Majority of individuals of the species under natural conditions, under observation. So now gene A has two alleles in a diploid individual. Big A makes a normal functional enzyme. The gene big A will make a normal enzyme. This normal enzyme will take this and convert it into a product. Little a will make a mutant enzyme, which is not functional as an enzyme. It cannot make the product from the substrate. It's still, protein is being made, the enzyme is being made, but it just can't convert this substrate to product. So, when we look at dominant relationships, big A, you're making 2 units of enzyme, making twice as much enzyme, and you're going to have this should be wrinkled. Okay? This should be wrinkled right here. My mistake. When you're big A, little a, this should also be wrinkled. Oh, golly. You know what? I'll tell you what, I'm going to make it easier. All right? Make sense now? You go from wrinkled to smooth. So when you're big A, you're making 2 units of this enzyme. And when you're making 2 units of the enzyme, you generate a product that's smooth. When you're big A, little a, you're making 1 unit of enzyme that's active, but you're still smooth. And it's only when you're little a, that you're making no functional unit of enzyme and you are wrinkled. So when you're looking at reactions where you're talking about an enzyme, in most biological systems, under most conditions, notice I use most, never always, okay? Most conditions, the amount of enzyme in a cell is always in excess. And because the amount of enzyme in a cell is in excess, you can have half as much enzyme and you'll still have the same thing. That's the basis of dominance relationships for most things. All right? Fair enough? Question?
So what happens with the enzyme unit that produces the rainbow phenotype? What does it do?
It's basically present, but it just cannot convert product, a substrate to product, is the definition. So that's why it's just, it floats around, it tries to convert it, it doesn't, it lets go of the substrate, moves on to the next substrate molecule. It doesn't affect it to a vast degree. Okay? So for genes that code for an enzyme, question.
So when we have a wrinkled phenotype, is that saying that there's no present enzyme? So there's
The enzyme is present, it's just not working. The protein is made, and you'll see that in today's lecture. Proteins are always made from, most of the time they're made from genes, but they may be mutant, and so they cannot convert the substrate to the product. All right? So in functional definition, it's not being made. You could just as well not make it. Wouldn't have any different phenotype because it's not functional. Okay. So enzyme activity is not limiting, and so 1 unit of enzyme is sufficient for your normal function. You don't need 2 units of it. Okay, so dominant equals a functional enzyme, recessive equals a non-functional enzyme. So for most enzymes, this is how you would imagine dominant versus recessive traits. Okay, clear? All right. Now, we're going to look at a phenotype that is something we call incomplete dominance. This is not following Mendel's rules in the heterozygous individual. Okay. There are a number of variations on this. In a heterozygous individual, when you are looking at phenotypes, and this is very important, you're looking at phenotypes. We're going to now look at something called incomplete dominance. Incomplete dominance is the occurrence of an intermediate phenotype. So imagine you have 2 phenotypes, red and white. Okay? So imagine you have a substrate that's white. It gets converted into a product that's red. Right? White to red. This is, if you're homozygous mutant, you're white. If you're homozygous wild type, you're red. According to Mendel, the heterozygous individual should also be red, but you are not. What you end up with is something that looks pink. So how do you explain that? And this is known as incomplete dominance. Okay? The heterozygous individual exhibits a phenotype that's an intermediate between the 2 homozygotes. Okay? And a good example of this is the color of flowers in plants. Happens a lot. Intermediate phenotypes, petals of a plant. Okay? Okay, so if you take a white plant, cross it to a red plant, this is a true breeding plant. This is Mendel's original cross, not Mendel's, but cross following Mendel's rules. You start with a true breeding parent that's pure white, cross it to a true breeding parent that's pure red. In the F1, if Mendel's rules were being followed, the heterozygous would be either red or white, correct? But they're not. What you end up with are flowers that are pink. All of the F1 flowers are pink. Now if you take these pink flowers and cross them to themselves, you end up with the F2. But here the F2 is not 3/4 one and 1/4 the other. What you get is 1/4 red, 1/2 pink, and 1/4 white. It's a 1:2:1 ratio. Right? These 2 here are your heterozygous. So your genotype ratio of Mendel, 1:2:1, and the phenotype ratio are identical. So for incomplete dominance, the genotype ratio equals the phenotype ratio. And the heterozygous has an intermediate color compared to the 2 homozygotes. Okay? So how do you explain these results? How do you relate genotype to phenotype? There's another example here showing you the same cross. one thing I should point out, when you see something that looks like this, when you have to write the genotype of this cross, you don't use big C and little c. You use numbers, C1 and C2, or R1 and R2, or A1 and A2, okay? Because we don't know what's dominant and what's recessive, so we stop using that convention of uppercase for dominant and lowercase for recessive. So for incomplete dominance, you basically just give numbers to the two different alleles that you're looking at, 1 and 2, basically. So C1 and C2. Okay? So C1 is white, C2 is red, and C1, C2 is pink, basically. So in practice, incomplete dominance can lie anywhere along the phenotypic scale. It could be that this C1, C2 could be dark pink, or light pink, or intermediate, or right in the middle between red and white, but it's in a range, right? So it doesn't have to be precisely in one spot. So if you go back to that original example of height, A1 is 2 feet, A2 is 6 feet, A1, A2 could be anywhere between 2 feet and 6 feet. That's incomplete dominance. It could be 3 feet, it could be 4 feet, it could be 5 feet. Anywhere around it could be an incomplete dominance. So, the phenotype of the heterozygous is the key. That will tell you whether the trait is behaving as a recessive trait, dominant trait, or an incompletely dominant trait. So always focus on the heterozygous to figure out how the trait is behaving, whether it's dominant or recessive or not. All right? Okay. So how do you explain this? So I've made this little pattern myself, just to give you an idea about it. Okay, here is one state, red. Here is a state that's pink. Correct? Looks kind of pink from a distance, especially at the back. Am I right? But when you zoom in, there's pink, there's red. They're all made up of the same red and white dots. It's just the intensity of the dots that's different. Right? There are gaps in here, and so this appears more pink than this one. And that's how you imagine incomplete dominance to be. It's the amount of pigment that's being produced is intermediate between the two extremes in the plant. So here you're looking more at substrate and product, not at the enzyme. And substrate and product are limiting in biological systems. Enzymes are not. So if you're looking at enzyme activity with the same amount of product and substrate and food, you will probably not see anything. It will be like a classic Mendelian. But if you're looking at the actual production of the substrate, the gene that makes the substrate, the protein, then you will start to see these kinds of phenotypes. Here's another example of the same thing. You've probably seen these in carnations in the florist, where you can have two different kinds of carnations, red carnations and white carnations, and you can cross them, and you will get pink carnations. Now, if you take a heterozygous pink cross to a heterozygous pink, you will end up with a cross. So R1 is the allele for red pigment, R2 for no pigment, and the R1, R2 will be pink pigment. When substrate is reduced, it leads to incomplete. All right. Let's take a break here, and then we'll go on to codominance, which is another phenotype that we have. Yeah, that's something. Okay, so we talked about one intermediate phenotype, and that was incomplete dominance. We're now going to talk about another phenotype that's kind of that's intermediate in a sense, called codominance. And codominance is very common. Okay? This is another variation on the classic pattern of inheritance of phenotypes, and that's called codominance. Now, the surface of your red blood cells, right? All of you are familiar with red blood cells. They have antigens on them, correct? What are antigens? Certain proteins, right? And they are present on the surface. What's the surface of a blood cell? Is basically a lipid membrane, right? Lipid